Good morning, everyone. Our final topic is about secondary hemostasis and fibrinolysis. These two books are the sources of our discussion for today. First, let's talk about hemostasis. So, hemostasis is a complex physiologic process that keeps circulating blood in a fluid state. Physiologic process meaning this is a normal process in a normal functioning body. So, as you can see in the picture, when there is an injury that occurs, it produces a clot to stop the bleeding, and then it confines the clot to the site of injury. And finally, it would dissolve the clot as the wound heals. So, when the hemostasis systems are out of balance, hemorrhage or thrombosis, which is pathological clotting, can be life-threatening. There is an absence of a single plasma procoagulant. This may destine the individual to lifelong anatomic hemorrhage. Hemorrhage means bleeding from a damaged blood vessel. Okay, so it can also lead to chronic inflammation and transfusion dependence. So transfusion dependence would occur when an average of more than 2 units of blood is transfused every 28 days over a period of at least 3 months. So conversely, when there is an absence of a control protein, this would allow the coagulation to proceed unchecked. Okay, so as a result, there could be thrombosis, it could lead to stroke, pulmonary embolism, deep vein thrombosis, and cardiovascular events. It's important to understand the major systems involved in hemostasis. So these are the blood vessels, the platelets, and the plasma proteins. So this is essential for us to be able to interpret laboratory test results and to prevent, predict, diagnose, and manage these kinds of diseases. As an overview, hemostasis involves the interaction of vasoconstriction, platelet adhesion and aggregation, and coagulation enzyme activation to stop bleeding. Okay, so there are two types, the primary and the secondary hemostasis. So primary hemostasis would refer to the role of blood vessels and platelets in response to a vascular injury or to the desquamation of dying or damaged endothelial cells. So what happens here is that the blood vessels would contract to seal the wound or reduce the blood flow. So this is the process that we call vasoconstriction. And then the platelets become activated. They would adhere to the site of the injury, secrete the contents of their granules, and cause aggregation with the other platelets to form a platelet plug. So the two processes, the vasoconstriction and platelet plug formation, comprise the primary hemostasis. So this is initial, it's just rapid, and is a short-lived response to vessel damage. But for us to be able to control major bleeding in the long term, the plug must be reinforced by fibrin. So this is where secondary hemostasis would come in. So secondary hemostasis is the activation of a series of coagulation proteins in the plasma. So mostly these are serine proteases, and this would form a fibrin clot. So these proteins would circulate as inactive zymogens, or what we call proenzymes, that would only become activated during the process of coagulation. So once they are activated, they would form complexes that would activate other zymogens and then would generate thrombin. So what is thrombin? So it's an enzyme that would convert fibrinogen to a localized fibrin clot, which is what we need to reinforce the platelet plug. Okay, so the final event of the hemostasis is fibrinolysis, which is the gradual digestion and removal of the fibrin clot as the healing occurs. So here you can see a table just comparing primary and secondary hemostasis. So primary hemostasis is activated by desquamation and small injuries to blood vessels, while secondary hemostasis is activated by large injuries. Okay, so primary hemostasis would involve vascular intima and platelets, But it's also important to note that although it's associated with primary, um, it is also associated with secondary hemostasis. So all this system would interact in the early and late hemostatic events. Okay, so primary is rapid and short-lived, and then procoagulant substances exposed or released by damaged or activated endotheliuses. So in secondary hemostasis, this would again involve platelets and a series of coagulation proteins in the plasma, and this is delayed but is a long-term response, okay, to vessel damage. And ones that are exposed on cell membranes is the activator tissue factor. So plasma transports at least 16 procoagulants, also called coagulation factors. So nearly all of these glycoproteins are synthesized in the liver. So this table just shows the different factors, their names, their functions, molecular weight, half-life, and their mean plasma concentration. So this was formed in 1958 by the International Committee for the Standardization of the Nomenclature of the Blood Clotting Factors, and they were officially named the plasma procoagulants using Roman numerals. And this is in order of their initial description or discovery. So it's important to note that when a procoagulant becomes activated, a lowercase a appears behind the numeral. So for example, activated factor 2. So this would appear as 2 and then a lowercase a behind the numeral. Okay, so here we can see factor 1 is what we call fibrinogen. Factor 2 is prothrombin. And then the numeral 3 was uh, tissue thromboplastin. So the numeral 4 is identified as plasma cation calcium. And then the numeral 6 uh, was assigned to a procoagulant, but then it was later determined to be an activated factor 5, which is why factor 6 was withdrawn from the naming system and was not assigned a name. And then factor 8 is antihemophilic factor, which is a cofactor that circulates linked to a large carrier protein, the von Willebrand factor. And then we have prekallikrein, also called Fletcher factor, and the high molecular weight kininogen, HMWK, which is also called Fitzgerald factor. So they have never received Roman numerals since they belong to the kallikrein and kinin systems respectively. And then there are platelet phospholipids, particularly phosphatidylserine, which are still required for the coagulation process, but they were given no Roman numerals. So they are called collectively the platelet factor 3. So this is a diagram showing the plasma components of the coagulation system. So there are four categories. So first are what we call the zymogens. So 8 are enzymes that circulate in an inactive form. So namely, this is a prekallikrein, factor 12, 11, 9, 10, 7, prothrombin, and factor 13. While others are cofactors. So this bind, stabilize, and enhance the activity of their respective enzymes. So namely, this is the high molecular weight kininogen, tissue factor, factor 8, factor 5, protein Z, protein S, and thrombomodulin. So there are also glycoproteins that are that act as controls to regulate the coagulation process. So namely, this is the antithrombin, heparin cofactor 2, the tissue factor pathway inhibitor, protein C, alpha 2 macroglobulin, alpha 1 antitrypsin, and the protein Z dependent protease inhibitor. So this is the coagulation cascade. So this is a series of steps in response to bleeding caused by tissue injury. So each step would activate the next and would produce a blood clot. So for an overview, we have an intrinsic pathway and an extrinsic pathway. So both pathways will eventually lead to the formation of prothrombin activator, which is factor 10. So once factor 10 is activated, in the presence of calcium ions, it converts prothrombin to thrombin. So when there is sufficient quantity of thrombin, it then leads to the conversion of fibrinogen to fibrin. So initially, these fibrin are monomers and cannot form a stable clot. So in the presence of calcium ions and the fibrin stabilizing factor, which is factor 13, it acts on them and forms fibrin polymers. And this can lead to the formation of a clot by attracting platelets and phospholipids and then eventually forming a stable clot and forms a platelet plug and leads to hemostasis. So the main goal of secondary hemostasis is to generate the key thrombin enzyme and produce a localized thrombus called fibrin. So the plasma procoagulants may either be serine proteases or cofactors. This is except for factor 13, which is a transglutaminase. So, serine proteases, these are proteolytic enzymes of the trypsin family, and this include the procoagulants thrombin, the factor 2, factors 7, 9, 10, 11, 12, and pre-K. So each of these member has a reactive serial amino acid residue in its active site, and it acts on its substrate by hydrolyzing peptide bonds. So serine proteases are synthesized as inactive zymogens, and activation would occur when the zymogen is cleaved at one or more specific sites by the action of another protease during the coagulation process. So, the procoagulant cofactors that participate in the complex formation are tissue factor, um, soluble plasma factors 5, 8, and the high molecular weight kininogen, while the remaining components of the coagulation pathway are fibrinogen, factor 13, phospholipids, calcium, and the von Willebrand factor. So this is a figure showing a simplified coagulation pathway. Okay, so we can see that if there is an exposed tissue factor, then this would activate factor 7. So factor 7 would then activate factors 9 and 10. So the complex factor 9 and factor 7 would then activate factor 10. So the activated factor 10 and 5 complex would then activate prothrombin or factor 2. So the resulting thrombin would then cleave fibrinogen to form fibrin. And um, this would then activate factor 13 to stabilize the clot. So thrombin activates also factors 5, 8, 11, and platelets in a positive feedback loop. So in the upper right part of the figure, it just shows that exposure to negatively charged surfaces like bacterial cell membranes would activate the contact factors 12, prekallikrein, and the high molecular weight kininogen, which would then activate factor 11, and then the cycle would continue. So we have what we call the vitamin K dependent prothrombin group. So these are the factors that would require vitamin K to generate a functionally active protein. So namely, this is the prothrombin, factor 2, factor 7, 9, 10, and the regulatory proteins, protein C, protein S, and protein Z. So these are all vitamin K dependent. So these are also called the prothrombin group because of their structural resemblance to prothrombin. Since all of the 7 proteins have 10 to 12 glutamic acid units near their amino terminals. So all of this, except protein S and Z, are serine proteases when activated. S and Z are cofactors. So vitamin K is a quinone found in green leafy vegetables. So this is just a box showing the food sources high in vitamin K. So it's also important to note that vitamin K is produced by the intestinal organism Bacteroides fragilis and Escherichia coli. So we know that some factors are dependent on vitamin K to be able to be functionally active, which is why vitamin K injections are given to newborns since at birth they still have low levels of this important vitamin. So to provide protection against bleeding and also serious diseases like hemorrhagic disease of the newborn. So, vitamin K injections are given to newborns. So, there is also a mnemonic for vitamin K dependent cutting factors. So, 2 plus 7 is 9, not 10. But usually I just remember it by the year 1972. So, 1972, it's 10, 9, 7, 2. Vitamin K dependent procoagulants are essential for the assembly of three membrane complexes that would then lead to the generation of thrombin. So each complex is composed of a vitamin K dependent serine protease, its non enzyme cofactor, and calcium bound to the negatively charged phospholipid membranes of activated platelets or tissue factor bearing cells. So the initial complex, which is the extrinsic kinase, is composed of factor 7A and tissue factor, and it activates factor 9 and 10, which are components of the next two complexes, the intrinsic kinase and the prothrombinase, respectively. So, intrinsic kinase is composed of factor 9A and its cofactor 8A, and it also activates factor 10 much more efficiently than the tissue factor 7A complex. So prothrombinase is composed of factor 10A and its cofactor 5A. So this would convert prothrombin to thrombin in a process that releases thrombin and a fragment called prothrombin factor fragment 1.2. So prothrombin fragment 1.2 in plasma is a marker for thrombin generation. So next is the cofactors in hemostasis. So procoagulant cofactors are tissue factor, factor 5, factor 8, and the high molecular weight kininogen. So there are also coagulation control cofactors, which are thrombomodulin, protein S, and protein Z. So thrombomodulin is also a cofactor in control of fibrinolysis. So each cofactor would bind its particular serine protease. And then when bound to their cofactors, serine proteases gain stability and increase reactivity. So first, tissue factor is a transmembrane receptor for factor 7A, and it is found on extravascular cells such as fibroblasts and smooth muscle cells. But under normal conditions, it is not found on blood vessels. So other cofactors in hemostasis are factor 5 and factor 8. So these are soluble plasma proteins. So both are activated by thrombin and inactivated by protein C. So factor 5 is a glycoprotein circulating in plasma and is also present in platelet alpha granules. So during platelet activation and secretion, platelets release partially activated factor 5 at the site of injury. So factor 5 is a cofactor 210A in the prothrombinase complex in coagulation. So, factor 8 is a cofactor to factor 9, which together form the intrinsic kinase complex. So next is the high molecular weight kininogen, which is a cofactor to factor 12A and prekallikrein in the intrinsic contact factor complex. This is a mechanism for activating coagulation in conditions where foreign objects, such as mechanical heart valves or bacterial membranes, or high levels of inflammation are present. The next is thrombomodulin. It's a transmembrane protein expressed by vascular endothelial cells. This is a thrombin cofactor. So together, thrombomodulin and thrombin would activate protein C, which is a coagulation regulatory protein. Both protein S and protein C are cofactors in the regulation and control of coagulation. So protein S is a cofactor to protein C as well as the thrombin activatable fibrinolysis inhibitor, while protein Z is a cofactor to Z-dependent protease inhibitor. So next is factor 8. So this has a molecular mass of 260,000 Daltons. And this is produced primarily by hepatocytes, but also by microvascular endothelial cells in the lungs. Then pre-factor 8 is unstable in plasma, which is why it circulates bound to von Willebrand factor. So during coagulation, thrombin is the one responsible to cleave factor 8 from von Willebrand factor to be able to activate it. So factor 8 and factor 9, these are two plasma procoagulants whose production is governed by genes that are carried on the X chromosome. So if you have a factor 8 deficiency, this is what we call the hemophilia A. Factor 9 deficiency, um, what we call hemophilia B. So these are sex-linked disorders that occurs almost exclusively in males. So factor 8 may be a cofactor, but it is very important in hemostasis since patients with hemophilia A usually experience severe bleeding and symptoms. Factor 8 deteriorates more rapidly than the other coagulation factors in stored blood. So in a thawed component plasma, the factor 8 level drops to approximately 50% after 5 days. So treatment for hemophilia bleeding episodes would consist of replacement therapy transfused according to the 12-hour half-life of factor 8. So von Willebrand factor is a large multimeric glycoprotein that participates in platelet adhesion and transports the procoagulant factor 8. So this is composed of multiple subunits of 240,000 Daltons each. So the subunits are produced by endothelial cells and megakaryocytes. So von Willebrand factor molecules are stored in alpha granules in platelets and in Weibel Palade bodies in endothelial cells. So the molecules are released from storage into the plasma where they are normally degraded into smaller multimers by ADAMTS13, which is a von Willebrand factor cleaving protease. So this occurs in blood vessels with high shear stress. So in cases like TTP or thrombotic thrombocytopenic purpura, where there is an inherited or acquired defective ADAMTS13 enzyme activity, so this is associated with ultra-large von Willebrand factor multimers in plasma that would result in platelet aggregation and microvascular thrombosis. So von Willebrand factor has receptor sites for both platelets and collagen. So it helps to bind platelets to expose some subendothelial collagen during platelet adhesion. So especially in arteries and arterioles where the flow of blood is faster. So the primary platelet surface receptor for von Willebrand factor is the glycoprotein 1B95. And then the arginine glycine aspartic acid sequences in von Willebrand factor also bind a second platelet integrin, which is the glycoprotein 2B3A, during platelet aggregation. So a third site on the von Willebrand factor molecule binds collagen, and a fourth site binds the plasma procoagulant cofactor factor 8. So von Willebrand factor is decreased in von Willebrand disease. So this is a relatively common disorder that occurs in 1 to 2% of the general population. So because factor 8 depends on von Willebrand factor for stability, so it's expected that individuals with von Willebrand disease who have diminished von Willebrand factor also have diminished factor 8 activity levels. So patients usually have factor 8 levels decreased to hemorrhagic levels. This is less than 30%, only in severe cases of von Willebrand disease. So the level of von Willebrand factor can also vary in people according to their ABO blood type. So group O individuals have lower levels of von Willebrand factor than any other ABO groups. So von Willebrand factor is an acute phase protein, as is factor 8. So the levels are expected to increase during pregnancy, in cases of trauma, infections, and stress. So here is a table showing the different types of von Willebrand disease: type 1, type 2, type 3, their corresponding defects, mode of inheritance, and clinical manifestations. So next are the contact factors that are also called intrinsic accessory pathway proteins. And these are namely factor 12, the high molecular weight kininogen, and prekallikrein. So they are named as such because they are activated by contact with negatively charged foreign surfaces. So in the diagram, you can see that factor 12A transforms pre-K, which is a glycoprotein that circulates bound to high molecular weight kininogen, into its active form kallikrein, which then cleaves high molecular weight kininogen to form bradykinin. So factor 12 and pre-K, these are zymogens that are just activated to become serine proteases, while high molecular weight kininogen is a non-enzymatic cofactor. So now, the contact factor complex, which consists of high molecular weight kininogen, the pre-K, and factor 12, then activates factor 11. So factor 11A is an activator of factor 9. So in cases of deficiencies of factor 12, high molecular weight kininogen, or pre-K, this does not cause clinical bleeding disorders. However, these deficiencies would prolong laboratory tests and would necessitate investigations. Factor 12 is activated either in vitro or in vivo. So in vitro, this is by negatively charged surfaces such as non-siliconized glass, kaolin, or ellagic acid that is present in the PTT reagents. So in vivo, these are activated by foreign materials such as stents or valve prostheses, and may activate contact factors to cause thrombosis. So factor 11 is activated by contact factor complex and more significantly by thrombin during the coagulation that is generated from the tissue factor activation. So deficiencies of factor 11, which is what we call the Rosenthal syndrome, usually results in mild and variable bleeding. So next is thrombin, which is the main enzyme of the coagulation pathway. The primary function of thrombin is to cleave fibrinopeptides A and B from the alpha and beta chains of the fibrinogen molecule. This then triggers spontaneous fibrin polymerization. In addition, thrombin amplifies the coagulation mechanism by activating cofactors 5 and 8, factor 11 by a positive feedback mechanism. So thrombin can also activate factor 13, which then would form covalent bonds between the domains of the fibrin polymer to cross-link and stabilize the fibrin cut. So this also initiates aggregation of platelets. And thrombin therefore plays a role in coagulation and in platelet activation and in coagulation control, and also in controlling fibrinolysis. So because of its multiple autocatalytic function, it is considered the key protease of the coagulation pathway. So next is fibrinogen, which is the primary substrate of thrombin. So this converts soluble fibrinogen to an insoluble fibrin to be able to produce a clot. So fibrinogen is also essential for platelet aggregation because it links activated platelets through their GP2B3A platelet fibrinogen receptor. So this is synthesized in the liver with a normal plasma concentration of 200 to 400 milligrams per deciliter, which makes it the most concentrated of all the plasma procoagulants. So fibrinogen is also considered an acute phase reactant protein, wherein its level would rise in cases of inflammation, infection, and other stress conditions. So the platelet alpha granules absorb, transport, and release this abundant fibrinogen. Thrombin also activates factor 13. This is a heterodimer whose alpha subunit is produced mostly by megakaryocytes and monocytes, and whose beta subunit is produced in the liver. So factor 13A covalently cross-links fibrin polymers to form a stable insoluble fibrin clot. So this is the coagulation cascade. So it comprises of the intrinsic, the common, and the extrinsic pathway. So first, let's talk about the intrinsic pathway. So this begins with any factor that causes trauma to the blood cells or its exposure to collagen. So this causes the activation of factor 12. So once activated, it acts upon factor 11, causing its activation. And then activated factor 11 then acts upon factor 9 to form factor 9A. So, this process would require cofactors like high molecular weight kininogen and calcium ions. So, activated factor 9 in the presence of activated factor 8 combined with calcium ions then form the conversion of factor 10 to its active form. And then the activated factor 10 and factor 5 leads to the formation of prothrombin activator, which then acts on the prothrombin to form thrombin. So, once thrombin is formed, it then acts on fibrinogen to form a fibrin monomer. So, this monomer is not stable. It cannot form a clot. So, this is where the fibrin stabilizing factor, factor 13, forms covalent bonds between the fibrin monomers, and this leads to a formation of fibrin mesh. So this mesh would then attract phospholipids and form a stable blood clot, which then causes the hemostasis. So now let's talk about the extrinsic pathway. So this is any pathway that begins with tissue trauma or trauma to the vascular wall or extravascular tissue. So after this, it releases the tissue factor, also known as tissue thromboplastin. This would cause the conversion of factor 7 to its activated form. So, the tissue factor then combines with activated factor 7 and in the presence of calcium ions would then cause the conversion of factor 10 to its activated form. So here, after this, the events are almost the same as the one we discussed in the intrinsic pathway. So once factor 10 is activated, this is now in the common pathway. So in the presence of calcium ions and factor 5, they form the prothrombin activator. So the prothrombin activator then causes the conversion of prothrombin to thrombin. Then the thrombin acts on fibrinogen to again form the fibrin. So again, this is just a fibrin monomer, so it's not stable. So the fibrin stabilizing factor, which is factor 13, then form bonds between these monomers, which again leads to the formation of fibrin mesh, attracting phospholipids and platelets, forming a stable blood clot, leading to hemostasis. So there are inhibitors and their cofactors that regulate serine proteases and cofactors in the coagulation system. So their function is to provide feedback loops to maintain a complex and delicate balance between thrombosis and abnormal bleeding. So they ensure that coagulation is localized and is not a systemic response, and they prevent excessive clotting or thrombosis. So the principal regulators are the tissue factor pathway inhibitor, antithrombin, activated protein C, which is the endpoint of the protein C pathway. So this is just a summary of the coagulation regulatory proteins as well as their functions and other characteristics. So antithrombin was the first of the coagulation regulatory proteins to be identified and the first to be assayed routinely in the clinical hemostasis laboratory. So, antithrombin is a serine protease inhibitor, also called a SERPIN, that binds and neutralizes serine proteases. And this include thrombin, which is factor 2A, factors 9, 10, 11, 12, prekallikrein, and plasmin. So other members of the serpin family include heparin cofactor 2, which is a minor inhibitor of thrombin, protein Z-dependent protease inhibitor, protein C inhibitor, which inhibits factor 12A, 11A, and kallikrein, alpha-1 protease inhibitor, which inhibits thrombin factor 11A and kallikrein, alpha macroglobulin, which is a thrombin inhibitor and also kallikrein, and alpha 2 antiplasmin, and also the plasminogen activator inhibitor. So next, we have protein C and protein S. So both are natural inhibitors of coagulation, and their synthesis is dependent on vitamin K. So they are activated by thrombin in the presence of thrombomodulin, which is a cofactor, then cleaves factor 8 and factor 5. So both are inactivated by the protein C inhibitor. So on your right side, you can see a figure showing the protein C pathway. So when does this occur? So during coagulation, the thrombin would propagate the clot as it cleaves fibrinogen. It then activates factor 5, 8, 11, and 13. So in intact normal blood vessels, the thrombin would bind to thrombomodulin and then trigger an essential coagulation regulatory system, which we call the protein C anticoagulant system. So the protein C then would change the thrombin's function from a procoagulant enzyme to an anticoagulant. So there is a transmembrane protein we call the EC protein C receptor, APCR, that binds both protein C and APC, which is adjacent to the thrombomodulin thrombin complex. So after this, the APC will dissociate from the APCR and bind to its cofactor, which is a free plasma protein S. So the stabilized APC protein S complex hydrolyzes and then inactivates factor 5A and 8A, which then results to slowing or blocking the generation of thrombin. So protein S is a cofactor that binds and stabilizes APC. So this is synthesized in the liver and it circulates in the plasma in two forms. So about 40% of protein S is free, but 60% is bound to the complement control protein C4b binding protein. So it's important to note that bound protein S cannot participate in the protein C anticoagulant pathway. So tissue factor pathway inhibitor. This is a Kunitz type serine protease inhibitor and is the principal regulator of the tissue factor pathway. So what this does is that the Kunitz 2 domain binds to and inhibits factor 10A, while the Kunitz 1 binds to and inhibits factor 7A and tissue factor. So this tissue factor pathway inhibitor is synthesized primarily by endothelial cells and is also expressed on platelets. So fibrinolysis is the final stage of coagulation. So this begins a few hours after fibrin polymerization and cross-linking. So this is a systematic accelerating hydrolysis of fibrin by bound plasmin. So there are two activators of fibrinolysis. So we have tissue plasminogen activator and urokinase plasminogen activator. So both are released in response to inflammation and coagulation. So what they do is they activate the fibrin-bound plasminogen several hours after thrombus formation. And then this degrades the fibrin and restore normal blood flow during vascular repair. So there is a delicate balance between activators and inhibitors, since excessive fibrinolysis can cause bleeding due to premature clot lysis before wound healing is established, whereas if there is inadequate fibrinolysis, this can lead to extension and thrombosis. So this is a table showing the proteins of the fibrinolysis pathway. So first, we have plasminogen, which is inactive. Its active form is plasminin, and its function digests fibrin or fibrinogen. So next is the tissue plasminogen activator. So this is secreted by activated endothelium, and its function is to activate plasminogen. Next is the urokinase plasminogen activator, which is secreted by kidney cells and activates plasminogen. So we also have plasminogen activator inhibitor 1, which inhibits tissue plasminogen activator. Then alpha antiplasmin, which is part of the serpin group, which inhibits free plasmin. So lastly is the thrombin activatable fibrinolysis inhibitor, which suppresses fibrinolysis by removing the fibrin C terminal lysine binding sites, blocking TPA and plasminogen binding. So once plasmin cleaves fibrin, it produces a series of identifiable fibrin fragments, so namely X, Y, D, E, and DD. So the DD fragment, which is also called the D dimer, is composed of two D domains from separate fibrin molecules, cross-linked by the action of factor 13A. So fragments X, Y, D, and E are produced by digestion of either fibrin or fibrinogen by plasmin. But D-dimer is a specific product of digestion of cross-linked fibrin only and is therefore a marker of thrombosis and fibrinolysis. That is thrombin, factor 13A, and plasmin activation. So this is just a table showing the different procoagulants and their designated Roman numeral.